So it's a material safety expert, that's right. It's in fact because you don't have only extractables and leachables, but also biocompatibility and physicochemical certification or requirements behind. That's why at UCB we decided to call that material safety expert, just for the short explanation. So I'm very happy to be there and to share with you the approach of UCB in the frame of this new USP 665 and 1665, and it was interesting to see the risk assessment by the other pharmaceutical company this morning and also the risk assessment performed by the suppliers. And like this, you can have a view on the different risk assessment and evaluation that has been performed by different pharmaceutical companies and different people. So I will start by a short story. So in fact, health authorities are more and more scrutinizing material safety. So as I explained, meaning extractables, leachables, biocompats, and physicochemical requirements. They are not only focusing on the primary packaging or on the final filter, which is part of the regulatory files, but also they are focusing on the risk assessment itself for all the materials, plastic for sure, and polymeric materials that you can have in your manufacturing process. So in the next slide, you have some examples of health authorities requests that you could receive after submission, for example. So the first one is coming from Japan and Turkish authorities. And the question, it was not related to the extractables data report itself, but more on the fact that, okay, why are you using the extractable study coming from these suppliers? What is your operating condition? Could you explain us why this studies is applicable for you? And at the end also you can have some questions regarding the toxicological risk assessment. Like for example, could you explain the calculation of the permitted daily exposure of these components? So just to give you an idea of the request that you can receive. The second one is coming from the MEA, and it's more related to the risk assessment itself. So they want really that you explain your risk assessment for the drug substances manufacturing process. So it's not only focusing on the drug product manufacturing process, but also for the drug substance manufacturing process. And the last example is in the same way as for the MEA, is the FDA request. It's also the same. They want to have a view of your risk assessment for all the polymeric material that you can have in your manufacturing process. So meaning not only the bag, the tubing, but as you can see in bold, also fitting, gasket, membrane valve, and the, for sure, associated certification behind the risk that you identified in your assessment. I think that this involvement is really due to the new USP 665, 1665, but probably also due to the new draft of the China Pharmacopoeia exposed by Etienne and Marine this morning. So I will not expose to you again the USP 665, 1665, but I will just focus on what UCB highlighted in this new regulation. So this new regulation is really applicable to all of the products that we have at UCB. So it could be non-chemical entities, non-biological entities, protein recombinant, antibody drug combination, cells, and also gene therapy. It's applicable to all your polymeric and plastic materials that you can have in your manufacturing process in contact with the process stream. And then it's also applicable for DS and DP pharmaceutical product, but also the DP pharmaceutical product. So API, as explained this morning, is out of scope of this chapter due to the highly purification you can have to produce your APIs. So I would like to stress the fact that we need to be ready. So everything, as suppliers and also pharmaceutical company needs to be ready for 2026. So for that, we need to start from now for some people, but for sure it was a little bit too late, as explained by Etienne this morning, but try to be more close as possible for 2026. So they increase the scope. They also give some definition for the methodology and the risk assessment that you need to provide. And they increase the requirement for whatever the risk, you need at a minimum some extractable data, which was not the case in the past for the low-risk materials. So it's very important to notify that. But at the end, the objective of, let's say, all the regulations are the same, whatever the requirements. So they want that all the materials that you have in your manufacturing process are not reactive, additive, or absorptive. This is really the goal of all the regulation. Then, what can we do to be ready for 2026? I think that the best approach is to put in place, if it's not already the case, a methodology, an approach to be able to be ready for this due date. And then I will share with you the UCB approach to what we call that UCB writes the material safety risk assessment, in other words, extractables and leachables risk assessment. So the first things to do is really to identify all the product contact materials you can have in your manufacturing process. So at UCB we have UCB sites, but also the CMO site, which is in scope of our material safety risk assessment. The second step is to identify the risk level of the materials that you have in your manufacturing process by using a risk scoring calculation level that I will expose just after this slide. And then based on this risk scoring calculation level, you are able to identify the risk of your materials that could be low, medium, or high. Once you have identified this risk level, the objective is to contact each supplier in order to collect all the data we can. So meaning data sheets, validation guide, extractables guide. And then after that, we performed an assessment of all the documentation that we received from the suppliers, and also for sure internal documentation that we have in our own libraries, to verify that in fact the certificates that you received cover your routine condition. So sometimes it's not the case. You can receive a certification, for example, a biocompatibility certification for tubing, which is autoclaved. And then in your routine process, you have a gamma irradiated tubing. So in this case, the supplier was not able to provide us the, let's say, right certificates, and then we need to start studies to cover our own routine condition. So once that we have all the data, so the certification and the additional studies that we performed, we're able at the end to conclude normally that all the materials that are in contact with your manufacturing process are suitable for the intended use and then for the patient. So this is the dream of all the material safety extractables and leachables experts to have this conclusion. Now I come back to the risk scoring calculation. So to be able to calculate your risk scoring, you need to know what risk factors will in fact will affect your leachables propensity. And then we decided to use different approach to perform our own risk assessment by using the BPOC guidance and also the USP 1665 665 and also our own expertise. And then based on that, we identify different risk factors that can affect the likelihood of leaching, as you can see. So the three first risk factors that are exposure to temperature, exposure to the duration, and the process-fluid interaction are common to every methodology that we compared. So as I think it was explained this morning, more higher is your temperature, more higher you have the risk to have some components that can migrate into your product, and the same for the exposure to the duration. So the process-fluid interaction is in fact a mix of, let's say, pH and also of your chemical composition, let's say, of your process stream. So in fact, we use the USP 1665 information and also information that we have in the BPOC guidelines to establish this process-fluid interaction. If you have a highly organic solution, for sure you have more risk to have some leachables in your product. The first one is the surface to volume ratio that we discussed also this morning, that has been used by the BPOC guideline, and we decided at UCB to use it also directly in our risk evaluation matrix, as it used as risk mitigation factors as per the USP 1665. But based on the data that we perform internally, we are able to demonstrate that this surface to volume ratio have really an impact on the potential leachables that you can have in your manufacturing process. And the last one is the material sterilization pre-treatment. So this one is used as risk mitigation factors per USP 1665 and also per the BPOC guidelines, but also at UCB we decided to add it directly in your evaluation matrix because it's really in fact an important factor also regarding the fact if you are flushing or rinsing your materials, for sure you will decrease the quantity of potential leachable that you can have in your product. So now that we have all these risk factors that has been established, I will show you the risk scoring calculation that we are using at UCB. So the first step is for sure to score each risk factor, so exposure to the temperature, exposure to the duration, process-fluid interaction, surface to volume ratio, and method sterilization pre-treatment. Once you have scored these risk factors, we perform a sum of all these individual factors, and we multiply this value by the clearance factors, which is the likelihood of persisting in the process. In addition, you can have some risk mitigation factor, like mentioned by the USP 1665, which is the chemical composition of your materials, the administration route, if you know oral solution, for example, and also the clinical therapy, so if it's acute or chronic duration. At the end, we can translate the value that we obtain by this risk scoring calculation in risk categorization, so low, medium, or high. Then, just to show you, so at UCB, we decided that before the last purification step, all the materials that are in contact with your product are at low risk. So it's not only due to the clearance factors, but it's also due to what we call the product and process risk assessment that we perform at UCB. So for sure, it depends the amount of chromatography step that you can have in your process. If you don't have enough chromatography step, for sure, the risk scoring calculation will be applicable before the last UFDF step. But in this case, for the example, we use the risk scoring calculation after the last UFDF step. So now that you have this in mind, I propose to show you some examples of the manufacturing process materials, and we compared in fact three methodology: the UCB tool, the USP 1665, and the BPOC guidance. So here you are in the drug substance. So for the upstream part, when you can have, as you know, bag, tubing, connectors, a lot of materials, and then based on our own product and process risk assessment and the clearance factor, we're able to mention that these materials are considered at low risk. So, as mentioned by Etienne, it doesn't mean that we perform nothing. It just means that it is at low risk, but we have to prove for sure the suitability of these materials by certification, like, for example, biocompatibility certification. So, and USP 1665 and the BPOC categorize these materials at medium risk. So in the next slide, this is the downstream part for the drug substance as well. And we compared for sure the three same methodology. As you can see, with the BPOC guidance, we have in fact most of the materials that are at medium risk, except the last one, which is considered as high risk, which is the final container for the drug substances. We have, let's say, a good granulometry by using UCB tool and also the USP 665, and I will just show you an example to explain the difference between the both tools, so the USP 665 and the UCB tool. So which is the PVDF membrane. It is the filters for which we have categorized by UCB tool as medium risk, and the USP 665 put it in low risk. This is really due to the chemical components of the material, which is the PVDF. So with the UCB tool, we could decrease by this level by 1 and arrive at the same level by using the chemical components as well. But for that, we need to have scientific rationale and data to prove, in fact, these risk mitigation factors. So really the really difference that we have between the BPOC and the USP and the UCB tool is this chemical of component and risk mitigation factor, which is not the same. Yes, also another example is the connectors that you can see in row 5, I think. You have these connectors at medium risk as per UCB tool and high risk as per the USP 665 tools. It's really due to the method sterilization pretreatment. So the fact that you have a pretreatment of your materials and also the fact that you have some rinsing and flushing of your materials after, for example, the sterilization. Okay. And the last one, last example is for the drug product manufacturing. So you have two steps, so the towing and the pooling, and also at the end the fill and finish steps. So we also made a comparison between the three methodology. And as you can see, we have, let's say, better granulometry with the UCB tool than with the USP tool or the BPOC guidelines. For the BPOC, most of them are high and medium. For the USP, most of them are medium and low. And by the UCB tool, we have, let's say, everything low, medium, and also high risk. So if you go to the row 3, so for the valve membrane, for example, it has been considered as medium risk by UCB and USP 665 and high risk by the BPOC protocol. I think that in this case it's really due to the surface volume ratio that we have in our own assessment that give us this risk categorization for this valve membrane. The last one is a tubing, really used at the end of the process step. So after the final, let's say, surge tank, you have a pump and then your product will go directly in your primary packaging. So this tubing is really used at the end of the process step. And in our case, so with UCB tool, the risk categorization for this tubing is high, which is low as per the USP 665 tool. And it's also the same really due to the chemical composition of the tubing that has been used. And for sure, the surface volume ratio is very high because you are already at the end of the process. That's why it has been categorized at high risk. So for sure, the USP 665 and 1665 is less stringent than the UCB tool and the BPOC in this case, but really due to this chemical composition of the components that are part of the USP 665, that we decided to don't use it due to the fact that we don't have, let's say, it's quite difficult to have information from the suppliers about the chemical composition of the materials. And also, for sure, we need to write some scientific rationale to be able to add it in the future in the UCB tool. So now that we have determined this risk level identification of all the materials, as explained, we need to collect the information from the suppliers and to evaluate the certification that we receive. So I will show you some examples of certification and the best practice that we applied at UCB. So the first one is a certification that we received from a suppliers for a gasket. As you can see, you have, let's say, every certification. So biological in vitro, USP 87, USP 88. You have also the physical chemical certification, so 381. And then the first question that we have when we receive this documentation is, okay, what is the pretreatment of this material? As this test has been performed on what? Non-autoclave materials, gamma-radiated materials, or another E-beam, I don't know, materials. And then we don't know because it's not written. So it's not possible for us to assess and to evaluate if we are compliant or not with the regulation. So we need to contact the suppliers to have more information about that. This is something where we are facing information that are missing in the certification, not for all the suppliers, fortunately, but for some suppliers it's true that we don't have sufficient information. And then the second question that we need to know, it's okay, but we don't have the extraction condition that has been performed for the biocompatibility testing, for example. It could be 24 hours during 37 degrees, but in our process, we have a contact time of 72 hours. And in this case, with these certificates, we don't know. We need also to assess and to evaluate and to come back to the suppliers to have this information. So for sure, we have a package from the suppliers with some information, but it's not possible for us to evaluate and to assess if we are compliant enough. So this validation package is not sufficient enough for UCB to assess and to claim the compliance to the health authorities regulation. So the next one is the material certification for filters, which has been categorized at high risk per UCB tool. So as you can see, we have some results, so NVR, FTR, and we have also a table with some compound that has been identified. So in this case, it was bisphenol A that we discussed this morning, and polyacrylate ester. So it's just an extractor. So that's why we have a good validation package with the information regarding the pretreatment. So for the first question regarding the pretreatment, it's fine, we have everything. But for the extraction condition used, you have only water as a solvent, and our product is more organic, so it means that we don't have sufficient data in terms of extractables. And also the analytical method used are more, let's say, for scouting and low-risk materials, and VR tox, so it's not sufficient enough for UCB to assess with this data. Another point is regarding, maybe I missed that in the previous certificate, but I can maybe come back. Yeah, so the version of the USP that has been used. So for the gasket, as you can see, is an old version of the USP. So USP 13, and then we are not able with this kind of certificates to evaluate if we are still compliant with the current regulation. So we need first to come back to the suppliers to have more data, or request a statement of compliance, or have our own statements of compliance, but for that we need more information from the suppliers. Sorry, I forgot this point. Then for the filter, it's the same question. Is the data of the validation package is sufficient? In our case, it's not sufficient. We don't have enough data. We don't have GCMS data for these filters at high risk. We don't have the value of the bisphenol A that we found. So how can we perform a tox assessment if we don't have the information? It's quite impossible. And the last one is an example from a tubing. So it's a very good validation guide. You can see that we have, let's say, everything. We have the right solvent, we cover the pre-treatment, the temperature used was also fine, the analytical method used was also very good. But at the end, this validation package is not sufficient for UCB as well, sorry, because we have 35% of unknown, and this unknown exceed the permitted daily exposure. So meaning that you have a lot of components above the safety threshold in the tox assessments. So you have two choices: perform a new extractable studies or perform a leachable studies to be sure that the components that we identify as unknown, let's say, are less the safety threshold in the leachable study. So for sure, it's not the case of all the suppliers, but it's just to show you where we have, what we have. We could have health certificates, and also what is the issue that we have behind, and then to be, let's say, compliance with the it's not compliance, but to comply with the USP 665 and the different, let's say, requirements for the low risk, medium risk, or high risk. We need also the help of the suppliers in some case, because with this kind of certification, we cannot assess. And for sure, the supplier receive a lot of questions and a lot of requests from customers, for sure, because need to send, okay, could you give us the result? Could you give us? And it's a lot and a month huge of workload. But we need really to work all together, pharmaceutical company and suppliers, to be able to be ready for 2026. So I arrive at the end of the presentation. So really the short conclusion is that the material safety is really more scrutinized by health authorities. So receive a lot of questions from everything. Could be China, Japanese, Turkish, Health Canada, as mentioned by other people this morning. We need really to be careful in the certification that we receive from the suppliers, and then to verify and to assess everything. And for sure, we need to have a good approach and risk methodology to be able to be ready on time.